Today I'm going to give you a demonstration of Abortus bang ring test or milk ring test. In short, it is also called as ABRT or MRT. What are the indication of this test? As you know that antibodies are not only found in serum, but they can also be found in vaginal and uterine discharges, in the milk and semen of infected or exposed animals. And this test, milk ring test or ABRT, is used to detect the presence of antibodies in bulk milk samples from Brucella infected cow or buffalo herds. This particular test does not perform very well with sheep or goat milk samples, and depending upon the herd size, the volume of milk has to be taken. If the herd size is less than 150 animals, you take 1 ml pooled milk sample. 150 to 450 animals, 2 ml of pooled milk sample. 451 to 700 animals, 3 ml of pooled milk sample. This test has got advantages as you do not require blood or serum for this test to carry out, so it is very rapid and simple test to perform. And but the limitation is that the only lactating animals can be screened, and further it has got lower sensitivity and specificity issues. For screening individual animals, RBPT is preferred. So for this test you require colored Brucella abortus antigen and you require the milk samples. So here today we are going to screen 2 milk samples, one coming from herd A and another one collected from herd B. So these are pooled milk samples. This is your antigen, cherry red colored antigen stained with tetrazolium. You may have blue colored antigen that is usually stained with hematoxylin. But these are, whatever the color of the antigen, this is inactivated antigen of Brucella abortus. 2 tubes are required for 2 milk samples. We are performing 2 tests on herd A and herd B, so you require 2 glass tubes. And in order to pour in the reagents, you would require, for antigen you would require micropipette and microtips, and for milk you will require autopipetter and pipettes. So with the help of the pipetter and the pipette in 2 ml of pooled milk sample into tube A. Make sure to change the pipette before you pour in sample B or milk sample B. Taking a fresh pipette, in a similar fashion, pipette in 2 ml of milk sample and pour it into tube B. Then with the help of micropipette and sterilized microtips, add 30 microliter of antigen into tube A and then into tube B after changing the microtip. After adding the antigen, make sure that antigen and milk samples are mixed together properly by tilting the tube 4-5 times gently. And this is important so as to ensure the interaction between the antigen and antibodies, if any, present in the milk against Brucella abortus. Change the tip and add 30 microliter into the second tube. And mix it properly as well. Make sure you have gloves on and the goggles on, because these are biological sample and they could be infectious. After the mixing of these milk sample with antigen, we'll incubate them at 37 degrees centigrade in the water bath or incubator for 1 hour. And after 1 hour, we are going to read the result. So after incubation, you see on your left the result is negative, and on right the test is positive. So in positive, you see the formation of a ring at the top of the milk sample, and this ring formation happens because of antigen antibody interaction and binding of the complex with the fat globules and rising up to the top. And with the fat milk ring, this positive milk ring test, you see the colored antigen and antibody in the form of ring. Thank you.